So our next speaker is Seamus Donnelly from Dublin. Aberrant TLR3 activation and more aggressive clinical phenotype in IPF. And I just want to point out that one of the goals of the next three speakers and the reviewers will be to keep Jeffrey Laurent awake, which the first four speakers were not able to do. So, Seamus.
That's a challenge, particularly post-prandial. Thanks very much, Arnie. I'd like to also echo previous speakers and my thanks to Luke and the meeting co-organizers for the very kind invitation to address you this afternoon and share with you some of the work we've been doing in Dublin in relation to TLR3 activation, viruses, and IPF. Now, I'm a respiratory physician, or as they say in the States, a pulmonologist, and I occupy very much that translational medicine space. And what it means for me is that I'm in my clinic with patients with IPF or other patient groups, and I see a clinical unmet need, and I go back to my scientists in my lab or to collaborating scientists in Ireland or abroad, and we, hypothesis-driven, we try and address at the bench some of those clinical problems that I see in the clinic on an everyday basis. I've been involved as an inflammatory cell biologist for some years, so we've been translating our observation to acute and chronic respiratory diseases over the last few years. With regard to idiopathic pulmonary fibrosis, it's a very frustrating disease for patients, their families, and for clinicians. It has a significant clinical unmet need, and one of the things that we have noticed as clinicians for many years is that many patients with IPF trundle along with very little loss of pulmonary function and very little loss of functional disability until there's an acute exacerbation, and that marks a precipitous decline and deterioration, ultimately leading shortly to their death. And we know from previous published work that if we analyze samples taken at that first acute exacerbation or subsequent acute exacerbations, that we can find or detect various viruses, particularly related to the herpes family. So there's a lot of animal work, particularly from Bethany and Galen, God rest him, from Michigan, in relation to animal models and superimposing viral infection on top. And Bethany, in some beautiful work some years ago, showed in her FITSE model that if she adds a HV68 viral infection on top of that, she gets more aggressive fibrotic phenotype. So that led us to suggest that potentially repeated viral infection would predispose individual patients towards a more aggressive fibrotic response, and we speculated via TLR3 dependent process. Now, the TLR receptors, as you're aware, there's 10 of them. We all have 10 of them within our own human bodies. And the specific one, there is sort of a family of receptors that are, if you like, an early warning radar, that the early detection of pathogens, be they viral, be they bacterial, fungal, or whatever. So TLR3 is probably the most characterized with regard to viral recognition receptor. As viral entry to cells, its highest concentration is often in the endosomal compartment. But fibroblasts are a little unique in that they also have significant cell membrane or surface TLR3 expression. And the TLR3 receptor is a question mark. We have the extracellular domain, transmembrane, and intracellular domain. And for get activation of the TLR3 receptor, two of them have to come together around this viral double-stranded RNA to get activation of the TLR3 pathways and ultimately production of interferon and interferon-related cytokines. We looked at the TLR3 promoter and 10 tag SNPs, and cut a long story short, we're going to focus on this SNP called L412F. Now, L412F exists on the extracellular portion on the inner concave side of the TLR3 receptor, and it's just next door to a very important glycosylation site. And that glycosylation site, we believe, is very important for full dimerization to occur. So when you are homozygous, certainly for this SNP, you do not get full dimerization. You get a blunted TLR3 activation response and a blunted interferon response. So then, back to our hypothesis, repeated viral infection will predispose towards a more aggressive fibrotic response via this specific SNP, which leads to ultimately a blunted interferon response. So first of all, we looked at a European population of IPF patients, and we looked at wild-type CC versus TT, which is the homozygous variant. And we found over 12 months that there was a significant association with early mortality in those patients that exhibited this TT variant. The odds ratio for protection was 0.15, but an odds ratio greater than 7 for chances of dying within that 12-month period. In fact, that TT ratio identifies 80% of all the patients, just over 80% of all the patients that died within that first year. If we look at respiratory death, it's basically the same message, a little bit more significant statistically. The actual prevalence of the TT in the Euro population is 1 in 8 of IPA patients. So 12% of the patient population express the homozygous variant, 37% are heterozygous. So almost half the population has this variant on one of its alleles. And the other thing, months to death, we have full survival data on the European population. And we can see that the wild type, the average survival was 48 months to death from diagnosis. The TT variant was 24. So then we wanted a validation cohort, and we began a very fruitful and continuing collaboration with Intermune. And you remember the INSPIRE trial published in The Lancet some years ago, in which they looked at the effect of the potential of interferon gamma beta as a treatment in IPF. Essentially, in that trial, patients were offered the opportunity to also donate their DNA if they wished, and some took that opportunity and consented. So there is a cohort that Intermune have of DNA collected from this trial, and with a very impressive clinical database of 96 weeks follow-up. We were very interested in the placebo wing of that group, which is 130 patients. And the other point to mention is that our European population was all comers of IPF. Good, bad, or indifferent, mild, moderate, severe, they were all comers. But if you remember for the INSPIRE, it was mild to moderate. So they excluded at the beginning a lot of the severe patients that would have died within the first year. But however, with that aside, what we found was a significant association with a hazard ratio of 3.5 when we looked at the TT, CTs together. That's 49% of the population at 1 year and 2 years. Now, as I mentioned, they have a very impressive clinical database, which includes sequential pulmonary function parameters done up to 96 weeks. So we wondered, was there any association between this particular TLR3 SNP and, if you like, rate of change or delta FVC over time? Well, that's just a reinforcing. The CCs, the wild type in this study, died at 76 years of age. The CT-TTs died at 68 years of age, reinforcing the point that this polymorphism identifies an aggressive fibrotic clinical phenotype associated with early mortality. So with regard to the pulmonary function data, we can see over the 96 weeks an increasingly significant accelerated decline in FVC in those patients that were homozygous or heterozygous for this particular SNP. We collaborate, very fruitful collaborations with the University of Michigan, in particular Corey and Steve Kunkel. And between ourselves and Michigan, there are 14 VATS biopsies that we've grown primary cells up and we have down in liquid nitrogen. And we basically screened those cells because we wanted to identify homozygous defective cells versus wild-type cells, activate the TLR3 receptor and see, is there any difference phenotypically between these cells? So we found a number of the TT homozygous cells. We basically compared the TT versus the CC, and we activated the TLR3 receptor with its synthetic ligand poly-IC. Busy slide, but essentially on this side we got messenger RNA, on this side we got protein. We used as a readout for the NF-kappa beta system simply IL-8 message and protein, for the IR3 rantase, and this is interferon beta message in this down here. So essentially what you have is you have wild-type cells stimulated with increasing concentrations of poly-IC. You've got heterozygous and you've got homozygous defective cells. And you see quite clearly that with regard to IL-8, with regard to rantase, with regard to interferon beta, there's a significant blunted response in those cells exhibiting this homozygous polymorphism. If you look at functional effect, and this is simply a FACS analysis for late phase apoptosis. We'll look at the top right-hand corner. This is wild-type cells. As you increase the stimulation with poly-IC, you get an increased apoptotic up to 30% in 24 hours. In the defective cells, that is very much blunted. So you get a pro-proliferative. So the cells are blunted interferon, they're hanging around for longer, they're more proliferative, and we have other data to suggest that they secrete profibrotic cytokines, significantly enhanced profibrotic cytokine secretion. So what about the in vivo models? Well, we've done with Corey, we've done a bleomycin model on a TLR3 knockout background. And essentially what we find is that the TLR3 knockout mice at day 4 have enhanced IL-13, enhanced TGF-beta, enhanced hydroxyproline in the lysates, again supporting the human genetics data, supporting the human primary cell data, that this knockdown of TLR3, either complete in the mouse or functional in the humans, is associated with the capacity to have a more aggressive fibrotic phenotype. So then we wanted to know what would happen if we reconstituted the cells or the animal models with interferon beta. Would we bring them back to a wild-type phenotype? So essentially with regard to those primary lung fibroblasts, if you look at wild-type cells and you stimulate them with poly-IC, you get a nice induction of apoptosis. That apoptosis is blunted in the defective cells. So if you add recombinant interferon beta, you restore the cell with regard to apoptotic rate towards its wild-type phenotype. Now we're just halfway through our animal studies in which we're giving recombinant interferon beta to our bleomycin on a TLR3 knockdown background, but essentially we got some survival mortality data. Now normally what you have in our bleomycin is, our bleomycin model, our mice don't normally die over 21 days. But essentially in this model we're going to give recombinant interferon beta after the acute phase at day 11 and daily thereafter to 21 days. And essentially what we find in our bleomycin TLR3 knockout model is we get a mortality of 60 to 80% over that 21 days. And when you give interferon beta, you can abrogate that mortality to 20%. So instead of 80% mortality, we're getting 20% mortality. Now, as I say, we're only halfway through these animal experiments. We're going to analyze the lysates the next week or two, but certainly that's very exciting data. So what we have, we've got a subgroup of IPF patients, 12%, that are homozygous for this defect. This defect is a blunted interferon response that we're showing in vitro and in our animal models in vivo. As I remind you, it identifies 80% plus in our European cohort of all patients that died within 12 months. So the opportunity, for example, in clinical trials for study enrichment using this polymorphism exists. Or conversely, you can use this polymorphism to enrich a more milder variant by excluding those that are homozygous for this variant. We also bring up the tantalizing possibility that maybe we should target these individuals, these 12% of IPF patients, with recombinant interferon beta, with the aim of not curing the disease, but converting them from that aggressive dead in 1 year clinical phenotype to a more slowly progressive, maybe 2 to 3 to 4 to 5 years down the line, passing away. And I know Ganesh did, he and his colleagues did that trial of interferon beta in 2004, but that was all comers. We're suggesting a more focused 12% homozygous clinical trial and see what happens. So it just remains to thank everybody who helped and did all the work, but particularly Michelle and David in our lab, Michael Keane. Michael and I run the National Pulmonary Fibrosis Referral Centre in Dublin. A very exciting and fruitful collaboration with Intermune, thanks to Xiao Charlene, David, Scott, and particularly Carl Cosson, Corey and Steve at the University of Michigan, Nick Carani in Edinburgh, and Maura White in Sheffield. And also, very importantly, I'd like to thank our funders. And I'd also like to thank you for your attention, and he isn't asleep.
Thank you. Seamus, very, very nice combination of clinical and basic studies. Jeffrey.
Thank you. And Arnold Brody has been known to jump to conclusions incorrectly in the past. And I'd just like to point out, just because your eyes are closed, it doesn't mean you're necessarily asleep.
Yeah, I've heard that before.
So what you've raised, I mean, it is a lovely combination of cell work, in vivo studies and genetics that are focusing on one particular gene. So this challenge that we face of subgroups, just if you could expand on that a little, and have you looked for this polymorphism in other settings, and other respiratory settings, maybe even not a fibrotic respiratory setting?
Well, Toby brought it up this morning, the importance of, we're a little toe in the water now. This is the first, I think this is a very important, I think this is identifying a subgroup which we've been talking about this morning as important, and also potentially guiding us with regard to therapy. With regard to, is this IPF specific or is this more globally fibrotics relevant? What we've done is we've analyzed the DNA of a cohort of scleroderma patients, and we do not find a correlation. We have some exciting data in relation to sarcoidosis, which David O'Dwyer, and I'll encourage you to go to his poster tomorrow to have a read about, so I don't want to steal his thunder. But David can tell you about this exciting data in the sarcoidosis front. But scleroderma, no. Just in genetics analysis, no. So it is not global fibrosis. We've also looked at cardiac fibrosis, and we can't find a significant message. But certainly we are getting a significant message with IPF and also with sarcoidosis.
Great, thank you.
Jack Elias.
Jack Elias from Yale. 2 questions. One, thank you, in case I forgot who I was. And no, we don't need any more comments. Two questions. First one is, the people who died in the 12 months of follow-up who had the polymorphism, did they get infected with the virus? Was there a clinical scenario that suggested that, or were there cultures that suggested that they handled the virus any differently, or did you happen to get there through the virus concept, but that the virus concept may not be the way that this is actually playing out?
Both cohorts that we analysed wouldn't have that data to hand, and certainly the European cohort is from 10 years ago, and I know that that's not on the database, and I don't believe, although I can be corrected from Carl in Intimune, whether they have that data to hand, but I don't think so.
All right. And the second question is, in the last 5 or 10 years, a second major pathway, the Rig-like helicase pathway, has been described in all organs that is a major stimulator of type 1 interferons. What it does is it detects cytoplasmic virus rather than extracellular virus. Have you had a chance in any way to look at the integrity of the Rig-like helicase pathway, because that pathway is very active. If you put poly-IC into the lungs of a mouse, the major pathway that makes type 1 interferon in the mouse is actually the Rig-like helicase pathway and not the TOL3 pathway.
Yeah, that's a good point, but we haven't had a chance to look at that yet. Jack.
Hi, Jack Goldie. I'm confused about the data about your demonstration that in the homozygotes, the release of pro-inflammatory cytokines seems to be blunted.
Yeah.
So
that wasn't what we expected. We expected the interferon to be down, but not the IL-8 side, NF-kappa B side.
So are they less inflammatory and more destructive, or where's inflammation into this now?
Yeah. I don't know. That was a surprising result, and I don't know how to. It's a real result. I mean, you know, you and I work with primary human cells, and they're a bugger to work with. So when you get a significant result like that, it's real. But why that is, do they have a blunted inflammatory response? I don't know.
But you got the bleomycin model. What happens in the inflammatory phase in the bleomycin in the homozygous knockout mouse?
That's true. We haven't looked at that. We have the samples, but we haven't looked at that.
Yeah. One more, Moises.
Do you know which is the effect of this polymorphism in epithelial cells?
No, we don't know. But one would expect that if they're not, say, interferon being produced, simply put, it wants to kill the virus cells, so it's pro-apoptotic. It doesn't want a fibrous sheath protecting the virally infected cells, so it's anti-fibrotic. So I would expect similar results in epithelial cells, that they will have blunted interferon response, that they'll be pro-proliferative, and that they may have a more pro-fibrotic phenotype. But I haven't looked, but that's speculative. Thank you.